Bioequivalence for highly variable drugs and reference scaled average bioequivalence, that is RSABE. This is the topic for the video. You might have come across the terminology of bioequivalence or highly variable drugs and the RSABE. This video is regarding the bioequivalence for highly variable drugs. Bioequivalence is a comparative bioavailability evaluation and comparative evaluation of the pharmacokinetics of the two formulations. As we know that generic formulations are required to be bioequivalent to the reference formulation. Also, whenever there is a SUPAC change which requires proof or demonstration of the bioequivalence, that time this study is required. As per the requirement for generic approval, bioequivalence, stability, CMC and other requirements are there. Bioequivalence is a very important requirement. Bioequivalence testing becomes very difficult when the drug is highly variable. And if the drug is highly variable, then there are chances that the bioequivalence study fails. So, for highly variable drugs, reference scaled average bioequivalence study is done so that you can understand or applicant can understand and demonstrate the bioequivalence for the variable drugs. Drugs may be highly variable because of the inherent properties of pharmacokinetics, sometimes because of the metabolic kinetics or metabolic uh, variations. Sometimes the variations are related to the absorption or metabolism. Sometimes the subject size increase may not work for the highly variable drugs. That time the reference scaled bioequivalence study is done. Always understand that the highly variable drugs are required to be demonstrated for bioequivalence through the RSABE, and for generic drugs, if the reference formulation shows high variation or reference formulation is a highly variable drug, that time the generic applicant can use this approach. Let's start with the video. And bioequivalence is the study which ensures that the generic drug or the test formulation demonstrate comparability to a reference drug in terms of rate and extent of absorption. So as we know, rate of absorption is denoted by the Cmax and extent of absorption is AUC. This ensures a therapeutic equivalence, safety and efficacy. As the generic formulations are required to be therapeutically equivalent to the reference formulations, so bioequivalence study demonstrate that the generic formulation is safe and effective as or comparable to the reference formulation. Regulatory authorities mandate bioequivalence studies for generic drugs. If bioequivalence is not demonstrated, that time generic applicant cannot get the generic drug approval. So bioequivalence study is important for approval of the generic medicines which are cost effective or which are affordable, and they ensure the generic provide the same therapeutic effect as brand drug. Regulatory approvals require pharmacokinetic testing, and this is tested through the bioequivalence studies. So first is average bioequivalence approach. So this is a generalized approach, and in this reference formulation and the test formulation or generic formulation is tested through a standard bioequivalence method. The bioequivalence parameters are Cmax and AUC. Cmax is peak concentration, AUC is area under the concentration time curve, and the acceptance criteria for average bioequivalence approach is 90% confidence interval for Cmax and AUC must be within 80 to 125%. You can go through the playlist of this channel. You will find many videos on bioequivalence. So in those videos you can find what is 90% CR and why it is required to be within 80 to 125%. So the average bioequivalence study approach may not work with the drugs which are highly variable. And what is the meaning of highly variable drugs? If the formulation reference formulation shows high intra-subject variability with more than 30% of variation or coefficient of variation, that is percent CV, that time you can say that the drug is variable. Always understand that the if the reference formulation is highly variable, that time only you can go for the reference scaled average bioequivalence. Large fluctuations in the drug absorption rates make proving bioequivalence difficult. So variability may be because of the differences or fluctuation in the absorption. There may be some other things also which are related to the high variability. Conventional average bioequivalence methods or approaches doesn't work for highly variable drugs because these methods lack sufficient statistical power for the highly variable drugs. And if the drugs are highly variable, that time there are the chances for false negative tests. These are called as type 2 errors, that is false negatives, and these false negatives are common due to extreme pharmacokinetic variability with the highly variable drugs. So understand here that you can say the drug is highly variable when there are pharmacokinetic variabilities and the intra-subject variability is 30% or more. So what is a reference scaled average bioequivalence? Many of the professionals find it difficult to understand. RSABE address high variability in certain drugs. RSABE adjusts bioequivalence limit based on the within subject variability of the reference drug. This is the very important statement that RSABE approach adjusts bioequivalence limit based on the within subject variability of the reference drug. And this within subject variability means the variability in the same subject. RSABE approach allows for more flexible acceptance criteria while maintaining the therapeutic equivalence. The core understanding from RSABE is that you need to understand or evaluate the within subject variability of the reference drug, and then you can have a flexible acceptance criteria for the test product. Methodology of the RSABE. These bioequivalence limits are scaled based on the intra-subject standard deviation of the reference drug, that is denoted by SWR. I am not going in detail for the statistical part because it will make this video lengthy and it will it may be confusing. So just for making you understand, the bioequivalence limits are scaled in the RSABE methodology based on the intra-subject standard deviation of the reference. So a replicated crossover study design is used to measure the within subject variability. So how you can measure the within subject variability? That time you need to give the formulation to the same subject 2 times because the drug is variable. If you give only 1 time, like in a standard approach, you will get some result. If you once again administer it or give it to the same patient, you will get different results, and that is called as the variability. This is my understanding from the RSAB approach, and I am conveying you. If you have any question or doubt, you can comment. Acceptance range widens for highly variable drugs, improving study feasibility. That time you know that the reference formulation is variable, and you go for the replicated crossover study. Replicated crossover study may be 3-way or 4-way. That is also called as partial replicate and the full replicate. So that is the study design you can use. Differences between the RSA, AB and AB. AB is average bioequivalence. RSAB is reference scaled average bioequivalence. So bioequivalence limits for RSAB are scaled based on intra-subject variability, and bioequivalence limits of AB are fixed, that is 80 to 125%. Study design is a replicated crossover for RSAB, and for AB it is a standard 2-period crossover, or it may be a parallel study. Always understand that for RSAB parallel study cannot be done. Applicability is highly variable drugs with percent CV of more than 30% for RSAB. AB is for lower variability drugs. Regulatory acceptance for RSAB is provided by EMA, FDA for highly variable drugs. And AB approach is a standard approach for the low variable drugs. Now come to the regulatory guidelines. EMA guideline is there, which is applicable for drugs with high variability, more than 30% CV. Replicated crossover study is required for this, and Cmax limits can widen to 70 to 143% if justified. Now see to the FDA guidelines. Similar to EMA guideline, similar CV threshold is there, 30%. More than 30% CV will allow you to go for RSAB. Replicated crossover study is required, and FDA guideline uses a regulatory constant 0.25 for Cmax to scale limits. Case studies are there for RSAB, like tacrolimus is highly variable. Generic venlafaxine, it is approved through the RSAB approach. Benefits of RSAB. So as we understood from the video, you can use this approach for highly variable drugs. It provides you the uh, enhanced statistical power, and that's why it reduces a risk of failing bioequivalence due to variability. Regulatory flexibility ensures fair assessment of highly variable drugs. It supports generic development, and that's why encourages development of affordable generic medicines to the public, and it ensures therapeutic equivalence. Though the Cmax limits are widened or reference scaling is done, but still it provides you the enhanced statistical power, and that's why it maintains patient safety and efficacy. Bioequivalence, in summary, is a requirement for the generic approval, and bioequivalence demonstration is difficult for highly variable drugs because there are many chances of the bioequivalence failure for the highly variable drugs with the standard average bioequivalence criteria or testing. RSAB provides a tailored approach for highly variable drugs bioequivalence studies, and regulatory bodies also support this approach. So you can adopt this approach if you know that the reference formulation is highly variable, and then you can ensure the bioequivalence is met. So this RSAB approach provides you a good way to demonstrate or prove the bioequivalence of highly variable drugs. Thank you for watching the video, and I request you to go through the bioequivalence playlist. Thank you.